first of all, let's talk about electron. It is a negatively charged particle of an atom that can be released by heat or electric field and only move freely in a vacuum space. Accelerated electron in vacuum space can act as light, but even better, because it has around 100,000 times smaller wavelengths compared to light itself. This moving electron, also called as photon, has less diffraction compared to light. The photon can produce a lot better magnifications in microscopy. Electron microscope comes in two types. One is called scanning electron microscope, or SEM, and the other one is called transmitting electron microscope, or TEM. Generally, an electron microscope contains an electron gun that produces electron beam, a magnification lens for magnification purpose, and also detectors to produce image. So first of all, let's talk about SEM. It is only be used to capture the surface of the specimen, and the specimen must be covered with coating. So this is what happens when the SEM is in use. The electron gun will produce the electron beam, and then the beam will go through the first lens, which is called the condenser lens, and then it will go through an objective lens that will focus the beam to go through a scanning coil, and then the beam will go and hit the coated surface of the specimen, and then the electron will be scattered, and these scattered electrons will be captured by electron detectors in the microscope, and these detectors will then produce a high resolution image of the surface. Next, for transmitting electron microscope, it can only examine non-living specimen, and also it only present 2D image of 3D specimen. Also, the specimen has to be dehydrated and also to be in ultra thin size, so it can be electron transparent, so the electron beam can pass through the specimen. So when the TEM is in use, the first thing that will happen is the electron gun will produce the electron beam, and then the beam will go through the first electromagnetic lens, which is the condenser lens, and then it will pass through the specimen at the specimen port, and from there it will go through the second lens, which is the objective lens, and then it will go through a projector lens that will project the electron beam onto a fluorescent screen, and then the image will be formed on the screen, and it will be captured by the camera under the fluorescent screen.
Application of electron microscope. First, in scientific research, second, in industry, and third, in forensic science. In scientific research, it is used to explore the molecular structure and mechanism of a particular disease. Second, it is to determine the structure of protein and to observe virus to provide cure to the disease. And the last one is to view the detailed 3D structure of biological tissue, cells, organelles, and macromolecular complex. In industry, electron microscope is used for high-resolution imaging to manufacture semiconductors. Second one, it is used in the modern industrial failure analysis as quality control tool. In forensic science, it is used to analyze the acute details of relevant specimens such as residue of gunshot, sample of blood, and clothing fibers. Second, to draw conclusion, identify material origin, and contribute to a body of evidence in criminal and legal matters.
Next, for the differences between light microscope and electron microscope. Application. For light microscope, have two applications, which is molecular imaging and cell imaging. For electron microscope, many biological using electron microscope to study about microorganisms. And the second one is electron microscope are often used in industry. Next is theory. For light microscope, are highly adaptable tools that can be used to analyze a wide range of specimens. For electron microscope, is a type of microscope that illuminates with a beam of accelerated electron. Last but not least is design. These are the differences design between light microscope and electron microscope.